Welcome back, guys. Welcome in another video lecture series. In this video lecture series, we will going to talk about different important life science experiments that is important from our CSIR net life science examination background. Okay, so today we will going to talk about Meselson's and Stahl's experiment. Okay, that tells us the kind of DNA replication, right? Whether the DNA is conservative, semi-conservative or dispersive. So first it is important to know about those three type of models. So there was a confusion what type of models DNA replication follows or how the DNA replicates, because we already know that DNA is double stranded helix, right? And DNA undergo replication, and it is quite clear that how they perform the replication. Okay, but there was a confusion that what in how they perform the replication, whether the replication consists of semi-conservative model or it is a conservative model or dispersive model. Because you know what, the Meselson style experiment was conducted in 1958, right? So in that time there was no sequencing or no anything that can give us a idea about how DNA replicates, right? So they there was near about three possible models that can be possible model for DNA replication. Okay, so the first model was the semi-conservative model. What semi-conservative model wants to tell us that after replication or during the replication, the parental strand breaks into two different single strand, okay, and a new strand, which I can say daughter strand, formed, which is exactly complementary to the parental strand. Okay, so that is what this picture want to tell us, right? And after that, and this is the condition after the first replication cycle. Similarly, after the second replication cycle, you will get four strand, and in each cases, first of all, parental DNA and the newly synthesized strand will broke, okay, and on the basis of that a new strand will be synthesized, right? And that is how it is going on. Okay, and that is the concept of semi-conservative model. Now I am not going to tell about that what model is the preferred one. We will going to derive it with the help of the Meselson style experiment. So then let's talk about the conservative model. This is another kind of model where the conservative model wants to tell us that the parental DNA will be intact, okay, but there will be a new double stranded DNA molecule, okay, and then in the second generation there will be again one parental DNA and one newly synthesized DNA will be synthesized from that one, okay, and there will be also two new DNA, okay, that is synthesized. Okay, that is the concept of conservative model. And in dispersive model, they want to tell us that parental DNA will be replicated into two daughter DNA molecule that contains a different proportion of parental DNA and daughter DNA. Okay, as you can see, there is a mixture. Okay, and this kind of model is known as dispersive model. Okay, now it is important to understand the components of the nucleotide, because we all know that DNA is made up of nucleotides. Okay, and nucleotides itself has three important parts. One is nitrogen bases sorry, and these nitrogen bases are of two types. One is purine for example adenine and guanine. We will not going to talk about their structure, we will talk about this in another videos. And second one is pyrimidine. Okay, those are thymine, cytosine, your uracil, although it is in case of your RNA. Okay, so that is the three. Those three are pyrimidine, and adenine and guanine is your purine. Okay, so here although I have taken a structure of purine, there can be a structure of pyrimidine. Okay, both are considered as nucleotide. Okay, and now there will be a pentose sugar. Pentose sugar means the sugar consisting of 5 carbons, right? Now, depending on the presence of OH group in the 2' position, because remember this carbon is known as 1', this carbon is known as 2'. So if there is a OH group in that position, then the nucleotide will be for RNA. Okay, and the nucleotide or the DNA which consists of nucleotide that contains a 2' OH group in its pentose sugar is not a DNA. Instead, it is a RNA molecule. Okay, remember it very carefully, that depending on what is present at the 2' end of the pentose of the nucleotide, it is distinguished or it is determined whether it is DNA or RNA. Okay, and the third component is phosphate group. Okay, so now try to think about this, how living cells get. So as here we are going to use prokaryotic E. coli. So my question is, how E. coli gets nutrients to make those nitrogen bases? E. coli simply gets it from the medium that we have provided, right? Now try to think about this. If we use a medium that contains N15 as the nitrogen source, what will happen? And we are growing that E. coli in that particular N15 nitrogen source containing medium for a number of generation. What will happen? The nitrogen base of that E. coli will have N15 nitrogen, which is not a usual one, because usually the nitrogen is N14. Okay, but in that particular cases, the E. coli DNA nitrogen base will having heavy nitrogen. Clear? So that is the basis of the Meselson-Stahl experiment. Okay, so let's talk about what they actually perform. Okay, so this is a picture of Meselson-Stahl experiment that I have taken from the internet. So what they have done, let's try to talk about this. First of all, they have grown the E. coli cells in a, so although they have provided us here, so they have grown the E. coli cells in a N15 containing medium. Okay, now the question is, what will be the new source from which we can get N15 or N14? So here the source is ammonium chloride. Okay, ammonium chloride containing heavy nitrogen in that case. So now if we grow E. coli cultures in N15 containing medium for a number of generation, and then if we perform cesium chloride density gradient centrifugation, we will get one band. Now let me give some basic knowledge about the cesium chloride density gradient centrifugation. Although I have made a separate video on how DNA can be separated with the help of cesium chloride density gradient centrifugation. So what is the idea of cesium chloride density gradient centrifugation? So it is a kind of isopycnic centrifugation where we are separating our molecule on the basis of their differences in Bowen density. Okay, now the question is, and what is the density gradient? So In density gradient centrifugation, we are separating our molecule in a medium that contains a density of gradient. Okay, and the density is greater from the top to bottom. Okay, what I mean to say that at the top of the tube the density will be lowest, and at the bottom the density will be highest. Okay, so in this density gradient centrifugation, now the question is why we are using cesium chloride. We are using cesium chloride because remember, at 1.6 to 1.8 gram per milliliter, the density of the cesium chloride will be equal to the density of the DNA. Okay, what does it mean? It simply means because the basis of the isopycnic centrifugation is DNA will stop migrating downwards when it gets a environment whose density is exactly equal to its Bowen density. Okay, and as a result it will form a band. Clear? It is kind a similar thing what we have discussed earlier in isoelectric focusing, where proteins stop its migration when protein gets a environment where the pH is exactly equal to its pi, and in that case, in the density gradient centrifugation or cesium chloride density gradient centrifugation, DNA will stop migrating downwards when it get a environment whose density is exactly equal to its Bowen density. Okay, and that simply means if our DNA is made up of heavier things, it will have a greater density. Okay, and as a result it will form the band at the bottom of the tube, right? And if our DNA is made up of some lighter things, then it would have a smaller density, right? And then it will give its band at the upper part of the tube. Clear? So that is the idea of the cesium chloride density gradient centrifugation. Why we are using cesium chloride density gradient centrifugation. Okay, so as here we are using a heavy nitrogen, so it is obvious we will get the band at the bottom of the tube. Clear? So now then scientists transfer that E. coli culture in a N14 medium. Okay, and here I want to give you another important concept, that for E. coli the replication, sorry, not it is replication, the generation time for E. coli is 20 minutes. That is the time required to complete one round of replication. Okay, so after that, after the 20 minutes in N14 containing medium, if we lyse E. coli cells and if we perform the density gradient centrifugation, that is cesium chloride density gradient centrifugation with the DNA that we have extracted from those E. coli, we will get a band that is not at that region that we have get at the previous one. Okay, instead this band is in quite a lighter region. Okay, so now up to that part it is quite clear that DNA replication is not conservative. Why? Let's try to think about this. If the DNA replication was conservative, then we will get two DNA molecules. One will be made up of 15N and another will be made up of 14N. Try to think about this, because let's say I am okay, so these two red lines are the two parental strand. Okay, now after first round of replication in 14 medium, we will get two blue strand, that is the newly synthesized strand, and two red strand, that is the your parental strand, if our DNA is conservative. But and if it happens, then in the tube we will get two band, because I have told you the cesium chloride density gradient centrifugation works by separating DNA on the basis of their Bowen density. Okay, and density is solely dependent on their mass, right? So it is obvious that N15 nitrogen containing DNA will have a higher density as compared to the N14, right? And as a result, in the test tube we will get two band, but here we have get one band. So it is clear that the DNA replication is not conservative. Okay, now, but now it is still not clear that whether it is dispersive or whether it is semi-conservative. Although this picture here, they have provided us what will be the DNA composition, but in reality we will get only photograph of DNA band. Okay, and just by looking on them, it is not possible to tell whether it is a semi-conservative DNA or dispersive DNA, and that is why scientists perform another kind of replication. Okay, by continuing the growing process of E. coli in the N14 medium. Okay, now let's look what they have get after the second generation or replication cycle 2. Okay, so here they have get two bands. Okay, one in exactly the same position that they have get after the replication cycle 1. Okay, and another one is in a lighter position. Okay, what does it mean? Now let's try to think about this. If our DNA is dispersive, then we will again get one band, isn't it? Because if you see this picture, the composition of these two band and the composition of these four band after second replication cycles are exactly same, right? So after the second generation of replication, we will again get one band in the test tube, but in reality we have get two band. That simply indicates us that our DNA replication is semi-conservative, because here there are two pure light strand DNA, right, and two pure hybrid DNA, right? Hybrid DNA means a mixture of parental DNA and daughter DNA, right? So that simply means our DNA replication is semi-conservative, isn't it? So after the replication cycle 2, it is now very clear to us that DNA replication is semi-conservative, and you can perform this experiment up to further steps, up to what you want, right? You can derive the what result after the replication cycle 10, replication cycle 11, replication cycle 12, but it is completely on you. But there can be MCQ question in your exam that after what replication cycle it was very clear that DNA replication is semi-conservative. So your answer should be after the replication cycle 2. Okay, so now let's move on. So this was the Meselson and Stahl experiment that tells us about that yes, DNA replication is semi-conservative. That means after the replication, the parental DNA, or during the replication, the parental DNA will be split into two different single-stranded DNA molecule, and on the basis of them, two newly synthesized strands will be synthesized, which are exactly complementary to the parental DNA strand, right? That is the semi-conservative model, and it was conducted, this experiment was conducted in 1958. Okay, and this was the explanation why we are using cesium chloride to make the density gradient, because at 1.6 to 1.8 gram per milliliter, the density of the cesium chloride will be exactly equal to the density of the DNA. So we can use this to perform the density gradient centrifugation. Okay, so here two important points to remember, that the parental generation or the parent E. coli, we have to grow the parent E. coli in N15 medium first, then in N14 medium. Okay, and the generation time for E. coli is 20 minutes. So I have given this information here because there can be a possibility that they will not give you the time. Okay, instead they can tell you, or sorry, they will not tell you that what round of replication they have performed. Instead they will tell you to write the number of light and number of heavy bands after 60 minutes. Okay, that simply means they want to know from you what will be the result after the third round of the replication. So you have to be very clear that the generation time for E. coli is 20 minutes. So now here I have provided a chart that will help you to remember about what will be the result. Okay, let's say first of all, after the 20 minute, that is the end of the generation 1, all will be hybrid, right? Because the parental DNA consisting of 2 heavy nitrogen will be divided into 2 strands. Okay, and each strand will having newly synthesized DNA molecule. Okay, and that is how we will get a hybrid DNA. Okay, so all will be the hybrid. Okay, so here we don't need to talk about the ratio. Now the number of the heavy strand here will obviously be 2, because here we will get 2 DNA, right? And each having 1 newly synthesized strand and 1 parental strand. So obviously there will be 2 heavy strand and 2 light strand. Okay, and after the 40 minutes, that is the generation 2, these 2 DNA molecule will be split again. Okay, and we will get 1 pure light double stranded DNA molecule and 1 hybrid. Okay, similar will be in this case. That simply means we will get 2 hybrid DNA molecule and 2 light DNA molecule, and that is why the ratio is here 1:1. Okay, and number of heavy strand will be, as I have told you, that there will be 2 hybrid. That means in 2 hybrid we will get 1 and 1 heavy strand. That means in total 2, right? And 2 pure light strand we will get, and 2 hybrid strand, right? So 2 plus 2 plus 2, total 6 number of the light strand we will get. I hope you can understood it, right? You can understand it, right? Because when this DNA will split, we will get 1 light strand, 1 pure light strand, and 1 heavy strand, just like that, right? So as a result, the number of the heavy strand will be this one, 2, and number of light strand will be 2, 4, 5, 6. That is why the 6, right? And after the time 60, that is the end of the third generation, we will get 2 hybrid, 2 light, with the ratio of, here I think I have made a mistake. Let's try to resolve it. So first of all, we have started our journey from parental strand, right? Then after the end of the first generation, we have get 2 hybrid, right? Isn't it? Then this is the end of the first generation, right? Then after that, after the second generation, we will get 2 DNA from this one, each having 1 hybrid and 1 light, and here we will also get 1 light, 1 hybrid, right? So that means 2 hybrid, 2 light. Okay, that means the second one, the result of the second one I have written wrong. That will be your sorry, and this is the end of the second generation. We are doing right. So then from that light we will get 2 light more, and from that we will get again 2 light more, right? And from hybrid, we will get one hybrid, one light, one hybrid, one light, right? So from here we are getting 2 light, and here we are getting 4 light. So 4 plus 2 will be the 6, right? So here we will get 6 light and 2 hybrid. Clear, my friends? So that will be the ratio of 1:3. And here the number of the heavy strand, so we are getting 2 hybrid, 2 light, and number of the heavy strand in the 2 hybrid DNA molecule will be 2, right? And the number of the light strand will be, we have get 6 pure light strand, right? So 6 into 2, that means 12, plus 2 hybrid, so there will be again 2 light strand, so 12 plus 2, that means 40. Okay, so that is how you have to solve the problems that will be in your examination. So let's see another problem. So before that, I want to mention another thing, that Meselson-Stahl experiment was the experiment that tell us that yes, DNA replication is semi-conservative in a prokaryotic system, right? So there are a similar experiment named Taylor's experiment that tell us that yes, DNA replication is semi-conservative in eukaryotic version. Okay, they have used Vicia fava for that experiment, although the aim of the experiment and the year in which it was conducted is exactly same, 1958. Okay friends, but this is the difference. Meselson used E. coli for their experiment and Taylor used Vicia fava for his experiment. Okay, and in that experiment he have used radioactive isotope, which was thymidine. Okay, so now let's move to, so another important difference between the Meselson and Taylor's experiment is, in Taylor's experiment we are using radioactive isotope. Please remember it very carefully. And in Meselson experiment we are not using any kind of radioactive things. Instead we are using N15, N14, but they are not radioactive. Okay, so now I have taken a numerical for you, that is, in Meselson-Stahl experiment, E. coli were grown on a heavy nitrogen and then transferred to light nitrogen. Here I want to mention one important thing. Please don't expect that all the time it will be heavy nitrogen first. That means, to check your intellect, the question setter can change the question in such a way that they will told you that in Meselson-Stahl experiment E. coli were grown on light nitrogen and then transferred to heavy nitrogen. So if it is, then please tell us what percentage of the DNA can be expected to be constituted of light nitrogen after the 5th generation of the multiplication. So entire calculation will be changed, right? So I want to mention you, or I want to suggest you that please try to understand the concept well. If you know the concept very well, then there will be no problem in any question that you will going to face. Although I will try to upload another video on Meselson-Stahl where we will going to solve MCQ problems from the Meselson-Stahl experiment. Okay, so please stay with us. We will surely try to launch another video on Meselson-Stahl where we will going to solve various kind of problems. So first of all, let's discuss this one. So in Meselson-Stahl experiment, E. coli were grown on heavy nitrogen. That means the initial makeup of E. coli DNA will be N15, N15. There is no doubt about this, right? And then transferred to light nitrogen. So after the first generation, we will get hybrid, right? We will get one N15, N14, another N15, N14. That is the situation after the ending of the first generation. Okay, then from in the second generation we will get one hybrid and one light, right? because N14 we will having another N14, and N15 will get another N14, right? Similarly here we will get also one heavy, one light. So this is the end of the first generation, this is the end of the second generation. Okay, now after the second generation we have one hybrid, one light, one hybrid, one light. Okay, so now we will again get one hybrid, one light, two light, one hybrid, one light, two light, right? So that means we are having 2, 3, 4, 5, 6 light nitrogen containing DNA and 2 heavy nitrogen, sorry, not heavy, 2 hybrid nitrogen containing DNA. Okay, and that is the situation after the ending of the third generation. Okay, so here we have talked about 5 generation, so we need to split it more. So hybrid will give us again one hybrid, one light, and from light we will get again 2 light, and from these 2 we will get 4 light, and here we will again get 4 light. Okay, and here we will get 2 light, and here in from the hybrid one we will get one hybrid, one light. So after the completion of the fourth generation we will get 1, 2, 3, 4, not 4, 1, 2, 3, 7, 8, 9, 10, 14 light DNA and 1, 2. That means 2 hybrid double stranded DNA and 14 light double stranded DNA. So this is the ending of the fourth generation, right? So now after the fifth generation the 14 light strand will be doubled, isn't it? So 28 light, and from 2 hybrid we will get what we will get. So from one hybrid we will get one hybrid, one light, and from one hybrid we will get one hybrid, one light. So there will be 2 light, 2 hybrid. So total 30 light we are getting out of 32. So 30 by 32 into 100 will be our answer. Okay. So our answer will be 215 plus so 25 into 50. So 25, if we will do 25, 55, so total is 37.5. So what we have getting, so the percentage will be 30 by 32 into 100, and this will be your answer. Okay, so I have taken a critical problem so that when you will face the problems having 3 generation, 2 generation, it will be a easier one for you. Okay, so I hope you have understood the concept of this very well, and thank you for listening to this class.